Right, so now we move into our final unit of the course, and that is ecology. And ecology is an incredibly broad topic. It is the scientific study of the interaction of organisms with their environment. So basically, where and how do organisms live? How do we understand how organisms interact with each other and their environment? Which other organisms are part of an individual's environment. So we've made our way, starting back at the beginning of the course, from atoms and molecules. We focused a lot on cells. We've looked at lots of different organisms, but now it's time to kind of turn the corner and look at more than one organism at a time. So let's review real quickly. Population is an interacting group of organisms of the same species, right? Community then is all the living components in a particular area, basically all the different species. And then finally, ecosystem is all the living and non-living components of a particular area. So you talk about the biotic factors, the living factors, and the abiotic, or the non-living factors, such as temperature, water, salinity, the amount of sunlight they get. What does the soil look like? What is the substrate? Is it sandy? Whatever. All those things are abiotic factors. Ultimately, we'll get to the highest ecosystem, or the most inclusive ecosystem, rather, and that is the biosphere, and that's the global ecosystem, right? So when you look at that global ecosystem, you start to see that things that happen in one area can affect other things very far away because everything's connected. So ecology then can kind of address many, many levels of this biology, starting with an organism. So looking at like an organismal ecology study would look at the physiology of an organism. Why does it have the traits that it does to interact with its environment or survive in its environment well? Like how does it occupy its niche? And we'll talk about that in just a little bit. To population level, community, ecosystem, you can talk about how ecosystems interact with each other, those borders, and then a global ecology. The key that kind of the heart of ecology that I want you to remember is this: it's an emphasis on interconnectedness, and that interconnectedness leads to complex systems, right? So think about modeling salinity in Chesapeake Bay. So you've got the ocean kind of on one end and freshwater running in from off the land on the other end. So you have a very interesting gradient of salinity, and how do organisms then inhabit those very different levels of salinity, or able to transverse and survive in different levels of salinity, knowing the problems that we know about osmosis, right? And if osmosis doesn't ring a bell, go back and review, because now's the time to start reviewing everything to see the big picture. The other thing that comes along with studying ecology, besides these very complex systems that are very difficult to model at times, is sampling, right? How do you sample then many individuals and get a representative picture to build a model? So oftentimes you don't take every individual, you can't count every tree in the forest, but if you take a sampling of them and then extrapolate from that sample and say these are representative of the bigger population, then you can start to build models. So that can be challenging. Like how do you do sampling of grasses in a field? Right? You take a small quadrant and you count those, and then you multiply it out. How do you sample polar bears in the Arctic? You tag them. In this case, they've tattooed it on the polar bear's gum, numbers that are unique, and then you go back and check it. I mean, very involved science, very hands-on science is ecological science. So the other thing to think about with ecology is, one, you have a huge breadth of tools. You have everything that you need. You need to know molecular biology. You need to understand computer modeling. You can use planes and satellites, all kinds of stuff. So it's a very challenging subdiscipline in biology. And then also the time frame is something to think about. When you study, for example, a riparian system or river system, riparian ecology, you have a cyclic annual cycle, right? So it's like you have a flooding season and then a dry season. And so you can get data points for 1 year. And how many data points do you need? You spend 5 years collecting data points, and then you realize that your experimental design was flawed. Oh, you got to start again. That's 5 years gone. Whereas in a molecular biology lab, you may do something over a few days and realize that you are going down the wrong path, and you can switch directions. So there are many challenges. Not all ecological studies happen over years, but again, it's something to think about. Now, let's talk about our first level of ecology at a population level, right? So population ecology is going to look at populations, right? So this is one level above organism in our hierarchy. And remember, populations are the level at which uh, evolution is really going to be seen, right? Because populations evolve through selection of fitness of the individuals. So we want to think about that we're now seeing the effects as populations change based on the selective pressure on the individuals. So if you wanted to study a population, what are the parameters that you can look at? So, if I said go across the street and look at Cal Anderson Park and study, give me some information about the squirrels who live there. What could you look for? Well, you could start to look at density and dispersion. Okay, how many they are and how they're dispersed within that area. Um, how many are alive and at what ages are they? So living individuals and their age distribution. We'll talk about survivorship curves when we talk about that. Carrying capacity, how many squirrels can that area support? What are the resources in that area to keep this population going? And then finally, life history. How often do they reproduce? How old do they have to be before they reproduce? How long do they care for their young? And we'll talk about K versus R strategies as we talk about this. So these are the parameters that we want to talk about. Again, this is a very, very general overview of population ecology. So again, when we talk about patterns of dispersion within a population's geographic range, there are three general patterns that we can come up with. A clumped pattern, think of schools of fish. A uniform pattern, penguins or Americans. It's like, give me an arm's worth of personal space, don't invade that. Or they're in a random pattern. And the random pattern we often see with plants because seed dispersal is oftentimes random, and then plants can't move, right? They're sessile organisms. So again, that first parameter is going to be density and dispersion. Density being the number of individuals per unit area, and then dispersion is the pattern of the individuals within that unit area. Now, the next idea, when we look at the number of individuals and their ages, actually, there are three major patterns that develop when you look at animals. And that is, it's called a survivorship curve, right? So these are idealized curves as examples, but again, it's a starting point then to compare a real population of plants or animals that you would study in the real world. And so a type 1 curve is characteristic of humans, where you have low infant mortality, high survivorship rates across many ages, and then as you reach kind of near the maximum age of lifespan, it drops off completely. So you'll notice that the x-axis is percentage of maximum lifespan. So that normalizes the curves because humans live a lot longer than squirrels, right, and clams. So converting that into a percentage allows you to compare these curves in a reasonable way. So squirrels then have a type 2 curve, and that is basically a straight line under these parameters indicates a constant death rate over the lifespan, right? So you're just as likely basically to find any given age to have an even distribution along the age distribution of any squirrel in a population. And then finally, a type 3 curve is what we see in many marine invertebrates, like oysters or sea anemones or barnacles, all kinds of stuff. And it's type 3 is high infant mortality, which then stabilizes until you reach a threshold age. So this is oftentimes what you see what we call broadcast spawning, where things in the ocean will release sperm and egg just into the water, and there'll be millions and millions and millions of sperm and egg. They find each other, you get zygotes, start to develop into larvae, lots and lots and lots of babies, but then the mortality rate of those babies is tremendously high. So you make lots and lots of babies, and then only a few survive. But if they do survive, if they get to some threshold age, then they're much, much more likely to survive for their entire lifespan. All right? Now, what this then suggests as we talk about this stuff about how many individuals you have in a population, it speaks to growth models, right? So when we talk about growth models, it's very important to think about that in an ecological setting because resources are almost always limiting. And what we see is two basic growth models, one which is really a component of the other, exponential growth, which you see the curve here, where you have a doubling potentially of the population with every generation, Or you can see what's called a logistic growth model, which is an idealized model for any given population as it approaches what's called K, or the carrying capacity. So you can see the big K there equals 1,500. So what you get is exponential growth that then tapers off into a flatline plateau where the population reaches its carrying capacity. So these are, again, idealized curves. So if you look at a paramecium population in a lab setting, if you provide it with a set amount of resources, what you see is the data points starting to scatter more to the top, but you do a best fit line, you definitely get that very, very idealized logistic growth pattern, right? So this logistic growth pattern just like that, right? You do see some scatter there because there is some kind of normalizing with the environment. Like there's death rates that increase and then decrease as birth rates go up and what have you, but basically it averages out to that line. And to speak to that, if you look at a different organism, the Daphnia, the water flea, under those same lab conditions, you get the same idealized best fit line, but you can see the data points actually average out a little differently. So you have this overshoot at the initial exponential growth that then has a crash that drops down below the carrying capacity and then now wavers up and down around that carrying capacity. And that's probably a little bit more realistic, even though this is still in a lab setting. Now, out in a natural habitat, you may get no growth curve that looks anything like an idealized model. Why? Because there's lots of random factors. Bad winter, drought, introduction of a predator. All these things get you a curve that looks something like the song sparrow population in natural habitat. It doesn't fit a nice, neat mathematical model. So do keep that in mind, and we'll come back to this idea a little bit later. Now, we've talked about this idea of density and dispersion, survivorship curves, type 1, 2, and 3, population growth models, either it's exponential, which ultimately leads to a logistic model when it reaches its carrying capacity. And so let's talk a little bit more about carrying capacity and what we call a life history. So when you talk about an organism's life history, you're thinking about how often it reproduces and how many babies it makes at each reproductive event, as well as how many resources it puts into those individual babies. So tequila is something people may be familiar with. And what you want to ask yourself is, what kind of plant does tequila come from? It comes from agave. So agave is a desert plant, and it's also known as the century plant because it sends up one flowering stalk, and it's on the order of decades. It's not really on the order of 100 years, but it seems that way sometimes. On the order of decades, it'll send up this one what's called an inflorescence. So it's a angiosperm, where it flowers, seeds, and then it's done. It dies. It puts all its energy into that one event, but produces lots and lots and lots of seeds. And so this kind of reproductive style is called Big Bang reproduction, and this is what you would incorporate into what's called the life history of an organism. All right. Now, what that then does is beg the question about those resources that you put into your offspring. And so what this suggests is a model that we call an R strategy versus K strategy with respect to population growth. So the R strategy is having lots and lots and lots of babies and putting zero resources into them. And so one extreme of that is like a clam, where you may have 500 million babies a year, egg output, and very, very little parental care. Versus primates on the other end, K, where you may have one baby every 5 years, okay, for chimpanzees. The baby of a chimpanzee, or for humans in that same sense, is very, we have to put a lot of resources into that child to make sure it hits the ground running, right? And so this is where the source of those names come from. So K, we already know, stands for carrying capacity. So that is the number of individuals that an environment can just support or carry, right? So that's just about how many resources we have, okay? Now, R is the symbol we use for death rate, right? So here, when we see an R strategy, the death rate is very, very high, but you're going to make lots and lots of babies, right, and put little resources into them. So I don't have to worry about expending too much energy, because I know that even if 0.1% of my babies survive, it's still plenty to keep the species going. Now, there's no such thing as like true R, true K. There's always a spectrum with their extremes. So things lie along the spectrum of how much resources you put into your offspring and the numbers that you have. You want to have a stronger K strategy in established environments, environments where competition is very, very high, such that you need those babies to be ready to compete as soon as they're independent. So that necessitates lots and lots of resources into the offspring, to the progeny, so that when it is ready to go independent, it can compete successfully. Our strategy oftentimes are seen in less competitive environments where resources are more abundant. You oftentimes see this in new environments. So like if you have like a volcano erupt or there's a landslide and you've just kind of eradicated all the species, you've eradicated all the competition. And so weeds and fast growing plants and animals that reproduce many in high numbers will populate that area first because competition is small and the progeny have little resources, so they need to utilize the resources of the environment very, very quickly with little competition. So this R versus K strategy is something to think about and is super important in population ecology. So again, this idea of populations is simple at first, but can get very complex very quickly. Now, again, very few populations follow that idealized curve, and even doing population ecology, it's hard not to stumble into the borders of community ecology, where you look at the interactions between individuals of different populations, right? And because that is going to affect the numbers of individuals. In this case, you can see predator and prey relationships with this up and down cycling of lynx and hare numbers as the snowshoe hare populations boom, the lynxes have plenty of food to eat, and then they overeat, population of snowshoe hare drops, and the food goes down, so the lynx population drops as well. So this is not unusual, but again, doesn't necessarily fit a nice, neat mathematical model of sustainability. Now, one population to consider that's important to us is the human population. And just to kind of leave you with something to think about, we look at the human population and think about what is the carrying capacity of this planet that we're on, right? Now, that population curve is clearly exponential. It is going up very, very quickly. The real question then is, are we on the part of the curve that like we see in the population model, that eventually we're going to curve off and slow down and reach our plateau? Or are we a data point on the Daphnia model that's way, way above the carrying capacity, and we're going to see a crash before we find that K for the planet for humans? And so we can talk about this more in class and in lab. But this is population ecology. We'll move on to community ecology next as we wrap up the quarter.